Hello, I'm Dr. Clare Craig, and I want to do a presentation for you today on how you can understand autistic spectrum disorder through one framework which links almost all of the risk factors, and this has major consequences for advice on folic acid supplementation. Before doing so, I just want to credit Dr. Tim Kelly, who had the idea for this framework and got me onto it, and I've just been digging into the evidence base and putting together papers and this presentation. So it's an important topic. Let's begin. The first thing I want to say is that this is a great case study for educating the public on public health disasters because it's got everything. It's got inadequate trials which are terminated early, that have minimal safety data and that did not use benign placebo groups. There are huge biases in the observational data. There is the malign influence of industry, which comes in at one point. And then we have the public health failure to protect the public because of wanting to do a greater good instead of thinking about individual harm. And there's been an inability to roll back bad policy. This framework brings together almost all of the ASD risk factors based on evidence from epidemiology, including the timing, genetics and molecular biology, and the pharmacological data for how drugs interact with this pathway. And once we've looked at all of that, we'll be able to come up with policy suggestions for improvement. Now, ASD has some unique challenges, largely around the diagnosis. So the definition of ASD has changed over time, and that makes it really difficult to measure the actual change of a disease. But people have done the work and they've figured out and split it up into different categories. And when you do that, you can see that there has been a genuine increase as well as a diagnostic one. This is just a quick overview of where we're going with this presentation. It's all about folate metabolism. All of the risk factors feed in one way or another to folate metabolism. But there's this odd paradox where high folate levels and low folate levels in blood are both risk factors. And when we've understood this, we'll be able to explain why that is. Now, folate is an essential ingredient for something called one-carbon metabolism. One-carbon metabolism is something that happens in every single cell. It's absolutely essential to life because what it does is it takes things like folate and turns them into slightly more complex molecules, which then feed into making the nucleotides that are needed for DNA and RNA synthesis and therefore cell division and DNA repair. But also it gives methyl groups needed for methylation of DNA, which is how we switch our genes on and off. So if a cell is changing what it's up to, it needs that methylation system to be working to say, let's do this now, let's do that now. What's critical to understand is that the brain is a particularly vulnerable organ with regards to folate because, especially in utero, almost all folate can only get in through active transport through something called folate receptor alpha, which sits at the choroid plexus, which makes your cerebrospinal fluid. So it pulls it out of the blood and pulls it into the cerebrospinal fluid, which has a higher concentration. So that's why it has to be active transport, whereas the other ways of moving folate are passive, and that can't work when you've got that gradient going the wrong way. All of the drugs we know that are risk factors for ASD interfere with this pathway. The genetic susceptibility relates to this pathway. And then crucially, folic acid, which is actually not a food as it's being sold, but a synthetic chemical, which our bodies struggle to metabolize, is also important to understand with regards to this pathway, because having struggled to metabolize it, we get a buildup of unmetabolized folic acid, and that can block the receptor directly, which competes then with our natural folate, so our cells get starved of natural folates. Now, there's a large subset of people who've got ASD who have autoantibodies to that folate receptor. So there's an immune element we need to talk about as well. And then many of the other risk factors feed into that immune pathway because they act as triggers setting off the autoimmunity. So first of all, let's just focus in on that folate receptor alpha and understand that a little bit better. So as I said, most tissues have got more than one way of getting their folate in because they've got these passive transport systems. And our brains do too. Once we're born, we start to use those more. But certainly in development, really the folate receptor alpha is the primary way in. And also in the placenta, folate receptor alpha is very important for getting folate to the fetus. Now, there are situations where that gene can be missing. And in a mouse model, if you remove that gene, it's catastrophic for neurodevelopment. They don't live. In humans, they do live. but there are severe neurodevelopmental complications, part of which includes an autistic-like phenotype. If you take those antibodies I mentioned to that folate receptor, which block the receptor, and you give it to a pregnant rodent, then that results in a phenotype of ASD as well. And recently it's been shown that this drug called folinic acid, which is similar to the kind of folates we have inside our cells, and can bypass that receptor if it's given at a high enough dose, that can provide symptomatic relief to children who've got ASD. And in rodent models, it can rescue the pregnancy, so that even if you've given the antibodies to block the receptor, you can use folinic acid to bypass it. Now, that won't translate to humans because rodents have got other ways of getting folinic acid in that humans don't have. This is where we're headed. I know this is a busy slide, but I just want to show you an overview, and we're going to build it up bit by bit. The key thing to know is that we're going to talk through 3 different routes into how that folate receptor alpha receptor can be targeted. And then we're going to talk about drugs and the genetics. Here's the first thing to understand, which is around what we do normally. So we have folate in the blood. And this folate in the blood gets attached to the folate receptor alpha at a cell surface, and that's folate receptor alpha pulls it inside the cell. And then those folates get used in 1-carbon metabolism, but actually it's only a tiny fraction of the folates that get used because we get less than 1% of our daily requirements from the diet. And if you have low levels in the blood, there are various things that happen to try to increase them. So the stores in the liver get released, and the same receptor, folate receptor alpha, gets upregulated in the kidney so that that receptor can start scavenging any folate that's in the urine so that it doesn't leave the body. So our bodies become much more efficient at hanging on to it if the levels are low in the blood. And then also we upregulate those folate receptors, so we have more of the receptors so that they can scavenge anything that's there to make it more efficient. So there are systems in place to keep everything in balance because it's such a vital thing to life. As I said, we only get less than 1% from our diet because after it's been used in DNA synthesis and repair, it gets recycled in various different ways. And 25% of that recycling goes through this enzyme, DHFR, dihydrofolate reductase. Now, what was found out in 2009 is that that enzyme, DHFR, is only 2% efficient in humans as it is in rats in the lab. So that is a serious bottleneck compared to rats, but all the safety work has been done on rats in terms of folic acid consumption. We've touched on people who've got problems with their folate receptor alpha, who have autistic spectrum disorder type symptoms. People who've got differences in that enzyme, DHFR, are also at higher risk. There's another enzyme further down the pathway, which is to do with the methylation of genes, turning them on and off, MTHFR. And people who've got changes in that gene, they also are at risk of ASD. And then finally, people who've got predisposition to autoimmune disease generally are also at higher risk, so they're more likely to end up with autoantibodies. Now, if we just focus on drugs, first of all, there's strong evidence of a link to ASD in women who take valproate, sodium valproate, which is taken for epilepsy. And that valproate drug interferes with the enzyme DHFR, and it also can interfere with the methylation pathway through MTHFR, and that's quite well established. Now, there are other drugs where there has been suggestion of an association with ASD, where the evidence is a little bit weaker, but the pathway that it would interact with makes sense. So first of all, we have paracetamol or Tylenol. Now, it's actually quite a big body of work on whether or not this has an association with ASD. And if you look more carefully at the studies where they have carefully tried to exclude confounders like fevers and infections and premature birth and maternal diabetes, when you do that, you start to see a stronger signal. And also when you look more closely at the 3rd trimester and in postnatal use, then you get a stronger signal there too. What happens with paracetamol is that when the body is using it, it produces a toxin called NAPQI, and the body's glutathione is used up trying to get rid of this neurotoxin. And glutathione otherwise is part of the folate recycling system and part of the folate methylation system. So if you're using it up, then you're going to be inhibiting the folate pathway. So there is definitely a hypothetical reason why you should be concerned about paracetamol. And then metformin is a drug that's given in diabetes, and metformin reduces levels of vitamin B12. And again, vitamin B12 is needed for the recycling of folates and for methylation that is part of that folate pathway. So the evidence for those two is not as strong as for valproate, but there's certainly concerns. Now I want to focus on synthetic folic acid. This has been around since the 1940s. It is sold as a dietary supplement as if it is just food, but the molecule is not the same molecule as natural folates. It's oxidized in more than one part of the molecule, creating a slightly different shape. Now, if you look at the folate receptor alpha there in the middle, what happens when it's got hold of some of this synthetic folic acid is it brings it into the cell. But because of its oxidated state, it actually has a very high affinity for the receptor, and the receptor can't let go of it very easily. So it's quite inefficiently released into the cell. And the remaining folic acid bound to that receptor is either destroyed along with the receptor in cell lysosomes, or some of it will get recycled back to the surface but still bound to the synthetic folic acid. So if it's still bound on the receptor is effectively blocked from any kind of natural folate use. And so when you've got a blocked receptor, what you find is you get a backlog in the blood of unmetabolized synthetic folic acid. And this unmetabolized synthetic folic acid actually looks like folate to the body, which means that the release from liver stores gets reduced and the renal scavenging gets reduced and the folate receptor alphas get down-regulated. So all the systems we have for protecting us when the folate levels are low work in reverse because the unmetabolized synthetic folic acid levels are high. And so that small amount of unmetabolized folic acid that gets into a cell then has to be reduced into a usable folate by dihydrofolate reductase because it's an oxidized form. So it's going into that enzyme, and whereas we saw that was only 2% as efficient as in rats, in humans as in rats, for natural folates, for synthetic folate, it's only 0.08% as efficient. So it's an incredible bottleneck. And importantly, different people have got different efficiency with this enzyme. There's a fivefold difference between different people. So some people are much more susceptible to having problems with this enzyme. Now, what we know in terms of ASD is that you can look for unmetabolized folic acid in the cord blood, and there is a relationship with higher levels and ASD risk, which is seen especially in black children. And babies who have fed formula milk are given very high levels of this synthetic folic acid. But you can get a formula milk that's got natural folates in it. And when babies are given the natural folate type of formula milk, they have much lower unmetabolized folic acid in their blood and much better folate stores in their body as a whole. So what you're having with this situation with synthetic folic acid is that in the blood, it looks great. But in the brain cells where it's needed, there's a sort of starvation situation. So it looks like a starvation in the midst of plenty. I don't think it's an either-or situation. I think probably when you've been eating synthetic folic acid, there'll be a period of time for which you're in the purple area. And then after that's been processed by your body, you'll be able to get to a situation where you're using natural folates again. What that means for most of our cells will be that they'd be stressed in the day and recover at night, because most of our cells can do that. But obviously there are cells, particularly in a developing brain, where they have to have folates accessible all of the time because it's a time critical process. So this graph just sort of gives an indication of that kind of idea that you've got periods where you've got very high unmetabolized folic acid and it's competing with the natural folates, but there's some recovery at night. It might be that those periods are much longer than that after each meal. There was one study where they looked at unmetabolized folic acid in Americans who had fasted for 10 hours, and they were too high in 40% of them even after 10 hours. So it's not just about vulnerable tissues that are reliant on the folate receptor alpha, like the brain. It's also about vulnerable time windows, which you have in neurodevelopment. So now let's look at that third track in, which was the autoimmune track. Now, critically, between 48 and up to 75% of children with ASD have got antibodies that block the folate receptor alpha. And that compares to only 0 to a maximum of about 5% of healthy children. So there's a big signal here that that autoimmune antibody is doing something with that folate receptor. In recent work, which has shown that folinic acid, which can bypass that receptor and get usable folate into the cell, does improve language and communication in children who've got those antibodies. So that proves that those antibodies, they're not just an incidental finding, they are causative, and the problem can be treated. Now, I'm not selling folinic acid as a treatment, because in order to get it into a brain, you have to have quite high doses of exposure to other cells in the body, and we don't have safety studies for it. And I think it's really important that we find out if it's safe before giving it to children every day for the rest of their life. And in my studies, they've shown that high synthetic folic acid during pregnancy will cause ASD, but high folinic acid does not. Now, I don't think that would translate to humans, because, as I said, rodents have other pathways in for folinic acid that human developing brains don't have. So if we just look at where these antibodies come from, I've kind of put that into a three-tier system. So it starts off with the capacity for forming antibodies against your own tissues, which is higher in people who've got predisposition to other autoimmune diseases. And it's also higher in people who've got obesity or diabetes, because they're just more at risk of that. And then the next step you need is something that looks foreign, that can be something that the antibody will attack. So there are situations where, for instance, bacterial infections can result in autoimmune disease because something on the bacteria that the immune response is reacting to and the antibodies are formed to looks very similar or mimics something that's a self-protein. So you get this molecular mimicry and a neoantigen being formed. But then to actually have the problem, you need something to trigger the immune response, because we've got systems in place in our body that says, don't attack that, it's you, it's yourself. So you have to overcome that. It's called breaking tolerance. So there's various ways in which that tolerance can be broken, and various risk factors which would increase the chance of that tolerance being broken. For example, vaccine adjuvants would increase that risk, as would general infection and inflammation. So all of those risk factors feed into an autoimmune pathway. And so just to go into a bit more detail, first of all, those antibodies can cross the placenta and get into the fetus. And there's two types of antibodies. There's what are called blocking antibodies, which block the receptor and stop it functioning, and binding antibodies, which bind to the receptor but don't stop it working. So it's worth understanding that difference. And then just to talk a bit more about these neoantigens, there is evidence that in a related condition, cerebral folate deficiency, children have antibodies to bovine milk protein, which looks very, very similar to folate receptor alpha. So those antibodies cross-react. And when those children are put on a milk-free diet, they get some improvements. So that's an example of something that, could be relevant here. And then in the dotted box there at the top, I just want to mention that there is a precedent for other synthetic vitamins, like vitamin B12, which is another synthetic chemical. When that's given, it can result in antibodies to the carrier protein that it binds to in the blood. So there's a precedent for autoantibodies being formed when you've got synthetic vitamins in place. Now, what's interesting about B12 is if you stop taking the synthetic B12, those antibodies go away. However, with mercury and with gold and with certain other drugs, what can happen is that you give those foreign things and they form the autoantibodies to a self-protein. And when you take those things away, the antibodies don't go away and the disease persists. So the implication is that giving synthetic folic acid could in itself be a risk for autoantibody formation. And then just one more point to make on this slide, which is that in celiac disease, there are sort of broadly two types of people. There are the people who've got the antibodies for celiac disease, and they also have gut damage. And those people end up on a gluten-free diet. And then there are people who've got the antibodies, but their gut is fine. And so they end up on a normal diet. And so those people with celiac disease and antibodies who've got a very much higher background risk, because they have a risk for autoimmune diseases anyway because of that, but the ones on the gluten-free diet have a fourfold lower risk of ASD than the ones who are taking in the kinds of products that have got synthetic folic acid in them. So this slide just brings it all together to show how those things all play into it. And I just wanted to put it up largely to come back to the drugs. So we mentioned paracetamol and Tylenol and how that interferes with the glutathione aspect of folates. but actually they are also associated with when people have taken a vaccine, or when there's been infection, or when there's been inflammation. So you've got a kind of confusion here where you don't know whether it's the vaccine that's a problem and people are taking paracetamol coincidentally and the paracetamol's innocent, or vice versa, or a bit of both. So there is that problem that needs to be unpicked. And then just cycling back to the vaccines, there is good evidence in many situations of a strong temporal correlation. So that's where the timing is very suspicious. But when people have looked at any one particular vaccine to see if that specific vaccine has caused an increase in ASD over time, they don't find a strong association. And that could be for a number of reasons, but one of the reasons could be that if the vaccine is acting as a trigger in somebody who's predisposed, then over time, once you follow them up, all of the ones that would have been susceptible have had some other trigger in the meantime, and so the measurement at the end is more similar. So I just want to put that out there, that's something that needs unpicking. Now, I think one thing that's important to understand is how the different parts of the brain are affected. And when you understand that it's about folate and about folate in the CSF, there's something interesting emerges, which is that the CSF covers the brain, and it also is inside the brain in these areas called ventricles. So those spaces on the brain on the right, that's bright blue on the diagram on the left. And so there are regions of the brain that are furthest from the CSF where the folate's coming in, because once the folate's in, it can get through cells, they can pass it along one to another, but the regions that are furthest away from the surface are the regions that are going to have the least folate accessible, and those are regions that are affected in ASD. So this pink region, the basal ganglia, the thalamus, the frontal areas here, and a lot of it, sometimes ASD is called a connectivity problem because parts of the brain aren't talking to each other as well. And you can see this is the paler area is where the white matter is, which is what's connecting. And of course, that area is all further away from the folate supply. Utero, the areas that are sensitive are the basal ganglia, the cerebellum, which you can't really see here, it's under here, which has its own little ventricle, but there's definitely areas in the cerebellum that are further away from the CSF supply, and the thalamus, and then these deeper layers of cortex. And that results in muscular problems with low tone, motor delays, and sensory abnormalities, as well as global disabilities. So that's the kind of most severe type phenotype. It also affects part of the brain that are quite close to CSF in the ventricles, so the amygdala and the hypothalamus. Now, that could be that because this isn't the only mechanism, but it could also be because those areas of the brain need stimulus from other bits that are being damaged. Then you have the regressive form that happens after birth. So some of this development has already happened, but language and speech can go wrong. And what's interesting is to sort of look at which bits of the brain are developing and which have the highest demand for folate at that time. And these children don't have the same problems with the tone and the gross motor delay, but they do have the other problems. And then there's this idea of a kind of later emerging or childhood ASD, where they say that they diagnosed it earlier, but it was always there. And there might be some truth in that, but there's also the potential that it's, again, different parts of the brain developing at that time. And so these children don't have the same motor problems. They don't have like terrible language problems, but their language doesn't advance the way that it should. And then just to put a very skeptical one at the bottom, which is the kind of adulthood. Now, in theory, what would happen if there was a problem that arose in adulthood? What would you see? So you'd see the parts of the brain that most needed folates just because they're metabolically active. And from that, you could deduce that you'd expect there to be mood problems, memory problems, and potentially kind of a brain fog type thing. And there is evidence that low folate causes depression and psychosis. I wouldn't call it ASD, but I just think it's an important one to add in there as a kind of comparison. Now, there's another important aspect to this, which is the timing of exposure. And a good analogy is to look at testosterone and the effect it has on particular character traits, so say competitiveness. So competitiveness is increased depending on testosterone exposure in utero, testosterone exposure in puberty, and also testosterone exposure on at any one day, any one time, to give a phenotype that is present at that moment. And I think you can think of this in the same way. There's an exposure in utero during brain development, at infancy, and also today. And that's why the leucovorin folinic acid treatment can affect the most recent exposure, today's exposure, but doesn't necessarily affect the previous one. So what is the epidemiology here? Because there are some studies that have claimed that synthetic folic acid in pregnancy reduces the risk of ASD. So I think it's important to kind of address that, and I think these studies are confounded. So if we just go for the really big studies, because there's lots of little ones, but if we just look at the really big studies, there are 7 of them, and 3 of them report sizable reduction, so up to 40% low risk. So they're making quite a bold claim there that you'd think you'd see in all the studies if it was true, but 4 showed no difference. And what's important is that, oddly, unplanned pregnancies have a much higher risk of ASD, 2.75 times higher risk. Now, that could be for all sorts of factors that we can only guess at. But what's important is that the difference was the same whether or not women were taking folic acid. So it wasn't to do with that. There's something else in addition that causes that big difference. But that big difference alone would create the illusion of a 48% reduction in ASD in women who were taking periconception folic acid because they were planning a pregnancy. So it's really important that you exclude for how planned the pregnancy was. And only one of these studies did adjust for pregnancy intention. And when they did that, they found there was no protection against ASD. In contrast, if we look at infants, it's very widely accepted that formula, which is heavily fortified with synthetic folic acid, is a risk for ASD compared to breastfeeding. So why do we have folic acid in our food? It's been put there to prevent something called a neural tube defect. So in development, early on, the brain tissue rolls up into a tube, and if you have an opening at the back end, the base, you end up with spina bifida, and at the head end, you end up with anencephaly or absence of a brain. So these two conditions are related to one another. And there was very convincing animal work that showed that the problem of neural tube defects, which they're called, was caused by low folate levels. So following that animal work, 4 randomized controlled trials were done in women who'd had a history of a neural tube defect before, and they gave them pharmacological doses of folic acid, and they did show a reduction in neural tube defects. Some of those trials were terminated early because of the one successful trial. There's never been any evidence that folate deficiency is what's causing the neural tube defects, because nobody's ever found a level of folate at which there is no risk. There is a risk at every level of folate. So this study in Ireland looked at different levels of folate and the risk in those women, and they found that the women who had the highest levels of folate had 4 times lower risk than the women with the lowest level of folate. So it's a gradient across the population. Now, what's tricky for public health is that 95% of neural tube defects occur in women who have no previous history. So you can't just target these high-risk women if you're trying to prevent neural tube defects. So there's only ever been a single randomized controlled trial in a healthy population of synthetic folic acid, and that was done in pregnant women or women who were planning a pregnancy, where they were given 800 micrograms a day, which is still a pharmacological dose. They had just over 2,000 women in each group, and they didn't have a benign placebo, and they didn't just give synthetic folic acid. So the group given treatment were given a multivitamin, and there were 6 extra neural tube defects in the placebo group. But what they also showed, and it was statistically significant, is that there were 9 fetal deaths for every prevented case. And if you've got fetal deaths, then of course you might just be seeing a survivorship bias, so that the same babies that would have had a neural tube defect had it so badly that they died. And so in the end, there weren't any neural tube defects to measure. But it wasn't just neural tube defects that were prevented. They found fewer of all types of congenital anomalies in this trial. But as I said, it wasn't just folic acid that was given to the women, it was a multivitamin. So that complicates things. And the placebo group were not given something benign. They were given copper, manganese, zinc, and vitamin C. And manganese is known to be something that can cause neurodevelopmental harm. So it's not benign. So we don't actually have any proper trial in a healthy population with a benign placebo group at all. And what they also found when they followed up these children is that there was a statistically significant rise in asthma, wheezy bronchitis, and eczema, which, if you look at the data that they've produced, would imply that on the balance of probabilities, a third of asthma and nearly half of eczema of the severe cases in infants in the US could be due to synthetic folic acid. They didn't follow up the women at all, the mothers, and they didn't follow up the children enough. But we do have some data on the follow-up. So following those studies, no more trials were done because they said it was unethical to do them. And so from then on, we were reliant on epidemiology to say, well, what happened? And the CDC did a work to show their success. And they looked at how many neural tube defects there had been from 1995 to 2011. But they only looked at live births. So this graph is live births of neural tube defect per 10,000 live births. So they didn't account for terminations. And between 1995 and sort of 2000-ish, there was quite a change in terminations because there was an increase in earlier screening at 12 weeks and there was better diagnostics diagnosing earlier neural tube defect. So there were more terminations. So you can't say that you've caused a decrease if you haven't measured those. They also didn't factor in that there'd been a change in the coding definition. So before the decrease, there was a situation where spina bifida had a big umbrella diagnosis and so had anencephaly, and then some of the more specific diagnoses within that umbrella were taken out and given their own codes. So the spina bifida code would have shrunk anyway, and the anencephaly code would have shrunk anyway, and they didn't account for that. And then finally, they ignored the fact that there had been a historically declining trend in the first place, which you can't really see on this graph, but if you go back far enough, it had been declining. So once they had done all of that, they then claimed that they had reduced the number by 28%, which amounts to 1,326 cases per year in 2011. Now you'll see on the graph that actually there's sort of two vertical dotted lines. One is where optional fortification happened and companies were putting it into breakfast cereals and the like. And then in 1998, they put it into all flour. And at that point, there wasn't really a reduction. The reduction happened before then. And what it works out as is that 235,000 US citizens were being exposed to synthetic folic acid in their food for a year for every prevented case of a neural tube defect, which I think is utterly disproportionate. Now, this is the data from Europe. So this is EuroCat data, where it shows live births, fetal deaths, and terminations. And you can see the orange bars get lower. So that's very similar to the CDC data, but they got lower because the blue terminations increased. And that line at the top of the graph shows the total. So the total just has not changed. So despite pregnancy supplementation, despite fortifications, breakfast cereals and the like, nothing changed. Now, we need to look at safety because that's really, really critical. And I think that we look at safety in drug trials the wrong way. So when we're looking at whether or not a drug works, it's reasonable to say we want to know with certainty, so we can use a measure of statistics that tells you beyond reasonable doubt that the drug will work. But when you're looking at safety, I don't think we should be looking beyond reasonable doubt that something is definitely unsafe. We should be asking, well, what about what's happening on the balance of probabilities? Because we don't want something that's likely to be unsafe just because we haven't proven it's unsafe. So to explain this better, I want to use an analogy of a dice. If you rolled a dice 10 times and you got 4 sixes and you ask the question, is the dice loaded? And we could do that to anybody and they would say, well, I need to roll it more times to really know. And that's exactly how we should be approaching, and that's how we should be approaching drug safety. But what a drug trial would say is, if you've rolled it 10 times and got 4 sixes, that would happen 7% of the time with normal dice, so we can ignore it. And I think a better interpretation is to say, well, we think it's unsafe, all drugs are unsafe, we just don't know in what way. And there's a 97% probability that if you're rolling a 6 4 times, then the dice is at least 10% loaded. And so if you look at it that way, you get a better feel, I think, for what's safe and what's unsafe. Anyway, I've looked at it both ways, to be fair. So this is a table which shows what the studies found. The statistically, I know it's a lot, but we'll go through it one row at a time. The statistically significant through a conventional measure, it says yes in that column, and then it's got a big blue arrow on the right-hand side. But I've put the others in as well. So we'll start with the top row. We've got otitis media at the age of 6, that's inflammation of the middle ear, and it was statistically significant. And they found that 12,944 children per 100,000, so that's 13% more children, had that condition if they were in the treatment group compared to the placebo group. So that works out as 8 children whose mothers took synthetic folic acid to have 1 child with a middle ear infection. For atopic dermatitis, that wasn't statistically significant by conventional means, but on the balance of probability, there's an 82% chance that it's at least 10% increased. And it worked out based on the data in the trial at 7,500, so 7.5% of children having a problem, or 1 in 14. For any allergy at the age of 2, it actually went the other way. So it looked protective. Again, it wasn't statistically significant, but it didn't all look bad. So I've put the things that looked good in as well. Now, the fatal death one's obviously critical. It was statistically significant, and it works out at 2.6% of pregnancies ending in a fetal death. And it meant that 39 women taking it would have a fetal death because of it. For congenital anomalies, as I said, it did protect against them as well. So for every 78 women taking it, a congenital anomaly was prevented of some kind. So that is a benefit. Now, there were too many multiple births, and that was statistically significant. 1 in 83 of the women had a multiple birth, and multiple births are not benign. So in fact, every single pregnancy complication, except those that are due to the baby being too big, every single one of them is more common in a multiple birth. So, if we're giving this to all these pregnant women, we can expect to be having way more obstetric complications than we otherwise would be. Now, there were more live births overall, and it worked out at 1 extra live birth per 38 women. And I think that's important to note, because you could argue that the fetal deaths are offset by the live births. And at a population level, that's true. At an individual level, I think you would find that hard to argue if you were talking to one of the mums that had lost their baby unnecessarily. Now, just to whip through the others, there were low birth weight was higher in the treatment group. Atopic dermatitis in infancy was, and that was statistically significant. There was an overall increase in serious and chronic disorders of any kind. At every stage when they did a follow-up, there had been more deaths in the treatment group than in the placebo group. It worked out about 1 in every 150. Asthma and wheezy bronchitis was more common, preterm birth was more common, and at the bottom there you've got neural tube defects. So 1 in 349 women had a neural tube defect prevented. So when you look at the whole picture, it's really not clear cut that benefit is outweighing the risk here at all. And obviously the sum you can do once you've got that data is to say, well, for every neural tube defect prevented, what was caused? And the ones in yellow are statistically significant. So on the same basis that you're claiming prevention, you could claim you've caused 45 cases of otitis media, 9 fetal deaths, 4 multiple births, 2 atopic dermatitis in infancy, plus all the other ones that didn't quite reach the conventional level for statistical significance, plus unknown maternal harm, because that wasn't even measured. Now, I've put a graph in here because I think it's important to show infant mortality in the first year of life. So the numbers on the y-axis are deaths per year in the USA. And you can see that there was a very, very predictable, tight decline from 1979 to 1997. And then in 1998, the year the flower was fortified, saw a deviation from that trend. The fact that there was a signal in the trials saying that there were more neonatal deaths, and then if you look at the population as a whole afterwards, there do seem to be more neonatal deaths, is I think something that really should have been highlighted and acted on far sooner. Now, overall, I think one thing that's important to understand that I didn't understand is that it's not a dietary deficiency in the way that it had been sold to me. So I thought the problem was that there was a small group of women who didn't have enough folate, and that if we all took some folate, those people would have enough and the rest of us would get rid of it because we don't need it. But that's not what happens. So there is no group of people with a sort of oddly low level. There's just a normal distribution in the population. And when synthetic acid, folic acid, is put in the food, the levels in everybody's blood increases. So this graph here is from Chile, but you can find similar graphs from other parts of the world. The dotted line shows what was happening before fortification and the solid line after fortification. So you can see there's only a tiny number of people afterwards who are in what was considered normal beforehand. And so what's happened is it's not really nutrition. It's a pharmaceutical effect. And in fact, the word nutraceutical, that combining of nutrition and pharmaceutical, was first coined with respect to synthetic folic acid. And what's important to understand is that we don't know what our bodies need. We don't know if there's a physiological reason why at particular times our bodies have lower levels of folate in the blood, or if there's certain conditions where you're better off having lower folate levels in the blood. So our body systems for getting this right for us have been hijacked by this pharmaceutical product, which is making it a high level for all of us all of the time. Now, so why did it end up in the food supply? And I think this is a really important part of the story. And it goes like this. It was first discovered in the 1930s and first sold in the 1940s. Pregnant women would have been taking it since the 1940s. And at the time, they didn't have a proper evidence base for guessing how much you needed. And 400 micrograms was an estimate that was put in as a recommended daily amount. But the US government made a real effort to calculate what was actually needed in 1989. They did a lot of work. They figured out that the body loses 100 micrograms per day, but has tons in the liver, 10,000 to 30,000 stored in the liver. So you don't need it every day. They also figured out that there was essentially two types, that animal products, so eggs and meat, have what's called 100% bioavailable folates, whereas fruit and veg is only 50% bioavailable because it has to be processed first in the liver. So they said, well, if you need 100 micrograms a day, but your main source is fruit and veg, we should say that the recommended daily amount is 200 micrograms a day. And that's what they did. So that was an evidence-based number. Then in 1998, the US government produced another report, but this time it was funded by multiple industry sponsors who had an interest in folic acid supplementation. And they managed to do that sum again. So they redid the bioavailability sum and said, well, you need to have twice as much if you're getting it from fruit and veg. So they turned the 200 into 400 and added in DFE, dietary folate equivalent. So they've doubled it for no good reason because that sum had already been done. But then on top of that, the companies took that 400 and said, well, we should give people 400 of synthetic product, whereas the synthetic product is 100% bioavailable. So what you're now getting is 400 micrograms of bioavailable product when you only needed 100 in the first place. And in doing these calculations, they've created a situation where it's essentially impossible to get what they claim you need from a healthy diet, whereas it's eminently possible for what you actually need. So here's what you get from food. I will just go through the table quickly. From plant foods, the best sources are spinach and beans, and I know not everyone eats them, but that if you did eat some of those, you're getting a huge amount which would top up your stores and cover you for the day easily. Then peanuts give you 71 bioavailable, asparagus 122, a cup of orange juice 101. So 1 cup of orange juice, you're done for the day. Now, animal products are sort of always thought to not really be a great source because the source is lower, but of course it's bioavailable. So 1 egg, 25, a bit of beef steak, similar, a cup of feta cheese, 48. So even if you were not really having fruit and vegetables at all, you're going to struggle not to get enough. And in fact, the people who don't have enough tend to have other problems going on. They tend to have alcoholism or malabsorption. And even if you look at populations who are starving, only a minority will have folate deficiency and anemia with it. So our bodies are great at holding on to it and our diets are adequate at providing it. But with the claim that we needed so much more, the entire daily amount, 400, which is obviously 4 times the actual daily amount, gets put into breakfast cereal But obviously people don't always have a single 30-gram dose, which is what the recommended daily amount is. People often eat more than that. So people are getting multiple times what they actually need in one single serving of breakfast cereal. Once it was put into flour, it was in the bread, in the pasta, in the pizzas, and you're getting big doses over the course of a day if you're now having a healthy diet in a population where it's in the flour. And if you add it all up, especially if you include a multivitamin, which would have your full 400, adults could be on 1,200 to 1,500 a day. So you're looking at 12 to 15 times actual daily requirements. Pregnant women are given supplements that are 800 micrograms in the US, so they have even more exposure. And formula-fed babies are receiving 2 to 5 times the daily amount. But I just want to emphasise that a normal diet would be completely adequate if the recommended daily amount was actually realistic. Now, I think it's important just to ask the question, well, what is needed for pregnancy? So you can do it from first principles and say, well, you know, we know roughly what pregnancy is about. So you need to make a baby, and a baby's got folate stores, and a baby's cells are made of folate. So how much folate is that? And you add all of that up, and it's up to 1.2 milligrams, which is 1,200 micrograms. And then you say, well, what about fetal turnover and losses and any inefficiency? So we can add a bit more on top for that, but that's being generous because the system's very efficient. Then the mother has to grow. The mother has to grow placenta and breasts and new blood tissue. And so you can add all of that in. And if you put it all together, you get to 2.9 milligrams to grow a baby. Now, some people argue, well, what about losses? You know, when you're growing, you might just be losing the folates along the way. But if you actually measure it, if you look at folates in the urine, then the levels are actually lower in pregnancy than in comparable adults because the pregnant women are holding on to their folate. So there isn't an argument that there's extra loss on top of what women would be losing anyway. So the total requirement to make a baby is maximum 2.9 milligrams, central estimate about 2 milligrams. And that works out at 11 micrograms a day of bioavailable folate, or 22 if you're getting it from fruit and veg. So it's eminently doable from food alone. But what's happening is that women are taking periconception supplements. They're told to take it for 3 months prior to pregnancy. Obviously, you don't always know when you're going to end up being pregnant, so it might be longer. But let's say a woman does take it for 3 months before getting pregnant and then has 9 months of pregnancy, often taking the supplement throughout the pregnancy, which is what women often do despite the advice that it's only for the periconception and first trimester. Then if you add up that whole year of supply, that works out with 800 micrograms a day at 292 milligrams. So it's 150 times what's actually needed for a pregnancy. Now, there are other health impacts beyond what we've seen in the children. So we saw that autoimmune diseases like asthma and wheeze and eczema was evident in the randomized controlled trials, and other observational studies have also shown that supplementation is related to those things. Folate has been shown to have a role in autoimmune thyroid disease, in type 1 diabetes, and in rheumatoid arthritis. There is a risk of higher levels of colorectal cancer and adenomas when people are given synthetic folic acid. That is the reason why in England, Public Health England never wanted to put it in the flour, because they knew about this colorectal cancer risk for the rest of the population. Prostate cancer might also be a risk with this drug. And it affects the brain. So folate status is related to depression, to schizophrenia, and to altered hormone responses, to polycystic ovaries, and to impaired fertility. So there's all sorts of potential negative impacts that have never been properly measured, which the entire population is exposed to when you're putting it into the food. So what we'd like to do in science, and what we can always say in science, is we want more evidence. You can always have more evidence, you can fill in the gaps, you can be more confident about certain aspects of things. And what we'd like to do in theory is have a randomized controlled trial wherein you could take pregnancy supplements and have one group with synthetic folic acid and one group with natural folates and see what difference it makes. But you can't do that because the whole population is being flooded with synthetic folic acid in the food. So you can't actually do the work. So I would argue it's unethical to be exposing an entire population to a synthetic chemical when we have evidence that it does cause measurable disruption of metabolism and that there are particular subgroups who are at extra risk of harm. So the first thing we need to do is to stop population-wide exposure. And we should be applying the same precautionary principle that you would apply when other chemicals show clear biological disruption. Now, of course, if we suggested that, there'd be a pushback. People would say, well, if we didn't do this, we would have more neural tube defects. Now, I think I've been clear that the data that that claim is based on is flawed. It just is flawed. But even if it were true, synthetic folic acid has not been properly tested for safety, and we're potentially trading a small reduction in these neural tube defects for a much larger burden of chronic disease. And of course, the burden of proof for safety for someone who's putting something into the food supply is with the people doing that, and they don't have the evidence base to say it's safe. So I want to just use Bradford Hill criteria at the end here to just show that this would be a reasonable thing to do. Bradford Hill criteria are the gold standard for medical causation, which was formed coming out of the evidence that smoking caused lung cancer. Because the thing is, there's no one thing that shows you that. It's lots of different things. It's looking at it from lots of different angles and producing the whole case. So this table goes through the whole case. The first column shows the evidence that we had in 1992 that folates were responsible for neural tube defects. And the second column is the evidence we have today that ASD is caused by synthetic folic acid in the food supply. So the first thing you need to say is, well, how strong is the association? Well, actually, for neural tube defects in 1992, the evidence was not that strong. There was no small group that had a risk factor. They couldn't get the correlation to show that measurements in the blood were related to risk. There is that evidence now. That study I showed from Ireland was very careful about the timing of the blood tests that they did in the first trimester. And so they did produce that sort of fourfold difference in risk between low level and high level. But that data was absent in 1992. For ASD, again, we don't have a brilliant measure, but you can see sort of hints at it. So in regions that have got high fortification, that's where you see high risk of ASD. And within countries like Russia, say, the rural population has a lower risk, and the population in Moscow that's more exposed to processed food has a higher risk. The same is true in South Korea, where Seoul has the higher risk, and they're the population that's most exposed to westernized food. Now, when studies have tried to measure it, the results have been in either direction. So I wouldn't say it's strong evidence by any means, but it is some evidence, unlike for neural tube defects. The next one on the list is consistency. So you want to say, well, are there exceptions that sort of disprove the rule? And for folate intake from the diet causing neural tube defects, the UK and Ireland was always a paradox because we have the highest rates in the world of neural tube defects, but there wasn't really a dietary problem. So even the people writing these trials and claiming that there was a problem would mention this and say that that was a bit odd, wasn't it? Now, for ASD and synthetic folic acid, there is no example of a country with very high synthetic folic acid in the diet, but low levels of ASD. So we don't have that counterexample. And then, of course, we've got the celiac disease example I mentioned earlier, where those on the fortified diet have got fourfold higher risk. Now, the next one is specificity, which is a strange one, really. So the idea is that if you want to prove smoking causes lung cancer, then you should have to prove that smoking causes only lung cancer. And if you show that smoking causes other cancers and also contributes to heart disease, etc., then that weakens your argument that smoking causes lung cancer. Now, I think that's a really odd one to have in here, but it's in here, so we're going to address it. It's similar to saying, well, if you've got something that will cause damage to the small blood vessels in the body, that should cause one disease condition, even though it's affecting every organ. And the same applies to a great extent for folates, because folates are needed in every single cell in the body. So if you've got a problem with folates, I don't think you should expect it to be one single condition. Anyway, for neural tube defects, they didn't have specificity because they showed all congenital anomalies were reduced in the group that took the multivitamin compared with the trace elements. For ASD, we don't really have specificity either. There's evidence that it causes multiple things, like the multiple births and the asthma and the wheezing, bronchitis, the colorectal and prostate cancer. So we don't have specificity. Then you've got temporality. So for ASD, we've got lovely temporality, where you have a date at which there's fortification in the 1990s, and after that, the incidence of ASD goes rocketing. So that's quite a strong piece of evidence that one thing happened and then there was a consequence of it. It's a bit harder to do that with neural tube defects because they're not claiming an intervention. But you can say, well, are there situations where there has been starvation and then you see a higher rate of neural tube defects? And so, for example, in the Netherlands in 1944 to 1945, there was starvation and there were higher neural tube defects as a result. There were also studies in California where women had been fasting in the first trimester and they had higher risk too. So there is some evidence there. Next, you have biological gradient, which is this idea that the more of something you have, the more risk there should be. And in 1992, there had been no clear dose response for folate levels and the risk of neural tube defects. Similarly, for ASD, the gradient between unmetabolized folic acid and synthetic folic acid exposure and ASD risk has never been properly shown. There are hints at it, like formula versus breast milk being a risk factor, but it's not really been properly measured. Next, we have plausibility. So that's the idea that it fits with what we know about science in that area. So for neural tube defects, we know folate's essential for DNA synthesis and cell replication and methylation, and the animal models backed it all up. Similarly for ASD, we know folate's central to neurodevelopment and genetics, and we also have animal models, so those are both strong. Then there is coherence. So is there anything that just doesn't fit from the science? For neural tube defects, we know there are genetic risk factors and animal models, and all of the mechanisms do fit. For ASD, again, we've got genetic risk factors that make the handling harder, that put people at more risk. We've got the antibodies that block folate receptor, we've got folinic acid rescue, so there is evidence for both. Then there's experimental evidence. Now, of course, for neural tube defects, before the public health decision, they had the trials. For ASD, there are these very small trials on folinic acid showing that if you bypass the receptor, you can get symptomatic relief. But there is no trial where it's been removed from the diet. And the thing is, we can't do trials because everybody's exposed all the time. And then you have analogy, which is, are there other things in medicine that act in a similar way? So other nutrient deficiencies like iodine also cause congenital malformation. So that's an analogy that works. For ASD, an analogy would be other synthetic vitamins like B12 can trigger autoantibodies. And in fact, neural tube defects themselves are an analogy because they show that folates are critical for brain development. And then finally, you want to show reversibility. So that evidence is absent for both at the time. So the point is that the evidence base overall is comparable or even stronger for ASD than it was for neural tube defects. So the idea that we don't have the evidence base to say we shouldn't be giving synthetic folic acid is just simply not true, because previous public health decisions were based on a weak evidence base that was weaker than that. So I think that gives a good summary of many of the risk factors, although we haven't addressed them all. For example, we haven't addressed why boys are affected more. But it's also worth noting that there can be confounders among these risk factors. So, for example, prematurity seems to be perhaps increased by synthetic folic acid, but it's also higher risk if you've got obesity or diabetes, although it does seem that those two factors have a risk over and above prematurity alone. But then, of course, premature babies, often they're born unable to suck, and so they get formula exposure. So that complicates things as well. Another complication there is that synthetic folic acid is linked to tongue ties, and babies with tongue ties are more likely to have formula exposure. So there's a risk that the in utero exposure is actually increasing the chances of exposure through formula. So there's a lot more work to do on this subject, but there is a huge amount of work out there already, and there are clearly numerous scientists who have a better understanding of specific areas of this than we do. So we're launching a collaborative project called folate.org. It's a website where scientists can come and share their findings and just create a big, broad evidence base from which we can demand removal from the food supply. And on that note, the UK government are planning to introduce it into UK flour, including organic flour. So on the 12th of December 2026, it will be illegal to put it into organic flour, but on the 13th of December 2026, it becomes mandatory. I would say on the basis of all of that folic acid was introduced on shaky evidence, and so it's completely justified to remove it on this much stronger evidence base, and that we should be reverting to the 1989 daily recommended amount of 100 micrograms of bioavailable folate per day for adults. We should introduce an upper limit for all synthetic supplements. There shouldn't be the amount that people are taking of these chemicals which hasn't been proved to be safe. I would suggest a daily upper limit of 200 micrograms of bioavailable folates in total. And actually that would be enough to cover pregnancy anyway, because of what we said, the needs of pregnancy are only small. And of course, as soon as you say the recommended daily amount is 100 micrograms, well, then everybody's getting it from their diet, incidentally, and the whole argument for fortification and supplementation would just disappear by default. So that's what I think we should be doing.